From Microbe TV, this is TWiP, This Week in Parasitism, episode 264, recorded on August 15, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone.
And because it's Friday, STD Friday, Daniel has a Giardia on his, or no, not Giardia. Wait, don't tell me. Trichomonas.
You got it. You did
T.
well.
T. vaginalis.
You did well. So good
I've
job
seen that
there.
one before.
Well, hello, everyone. I'm glad to be here. I wonder how you keep it all straight, the podcast all about, and I'm like thinking viruses, what are you going to talk about? So
sometimes I mess up. Yeah.
So
it's okay.
It's just the two of us today.
Oh, yes. Christine is, I don't know, where is at some meeting in some other country or something? Where is she?
She did tell me we should put not only Christina out, but we do on the schedule, right? For the clinicians I work with, if you're off, you've got to tell us what you're doing. So.
It's okay. We can handle it. We used to, we've done some before, you and I, right?
We certainly have. So I think we'll get through this.
Yeah, we have a paper to do today, right?
It's a great paper, actually. And I've got it I have two ways. I've got the paper copy with all kinds of, old fashioned, underlined with my pen. And I've got got the PDF up here on Acrobat. So, but let's jump into it, because I feel like this is a theme that I find it to be interesting, fun. There's an author that writes books down this road. The whole idea of parasites affecting the brains of those that they infect. And so we have the article today, Toxoplasma gondii, infection of neurons, alters the production and content of extracellular vesicles, directing astrocyte phenotype and contributing to the loss of GLT1 in the infected brain. Okay, and this is published in PLOS Pathogens. Should we take turns? Did you want to do the authors so that I don't stumble through all the mispronunciation?
We can. These authors are from the University of California, Riverside. We have Emily Tabai, who is the first author, Ziting Gao, Nala Kachur, Arzu Ulu, Stacy Gomez, Zoe Figueroa, Christina Bergerson, Wenwang Zhang, and Emma Wilson is the lead author on this one. Toxoplasma.
All right. So it's a very, I have to say, we'll start off with the high level. And the nice thing they have is you've got your abstract, but nowadays they also have these author summaries.
More things for us to write, right?
But I think sometimes it's good, because the abstract sometimes has sort of like a structure to it. And the author summary is really just kind of give me the high level, why do I care? Why should I read this?
Dixon used to say that Toxoplasma was the most successful parasite.
I'm going to agree with Dixon, and they start the first line of their introduction, which did not make it to the author summary. Nearly 1/3 of the world's population is infected with the obligate intracellular parasite Toxoplasma gondii. So 1 in 3 individuals.
How would you get infected, Daniel?
So there's a couple different ways, but really the way is somehow you've got to, in most cases, you've got to ingest this, right? So there's a couple different ways that can happen. One of the ways, interesting enough, is through undercooked meat, right? And so that's particularly a problem in a lot of parts of the world. But also there can be mother to child transmission as well.
Can get it from cats also, or something like that?
Well, the felines are really the definitive host, so they're often involved in the life cycle.
That's why pregnant women should not clean the cat litter, right?
If they're going to clean the cat litter, you've got to clean it like every day on a regular basis, because there is a period of time, like once it ends up in the cat litter, before it becomes infectious. So if you're cleaning it every single day, maybe that's okay. But yeah, in general, we say don't, because you might not get it all cleaned every day, and then you could get exposed that way from the cats. So yeah, I would agree. Very, very successful parasite. And as we think about this, when we talk about the fact that 1 in 3, of the world's population is infected, and that this might actually have some impact upon cognitive function and behavior, sort of interesting questions arise. So, author summary, we'll start with there, and then I'm going to go through a lot of sort of what I think are highlights for folks. So, infection with Toxoplasma gondii leads to these neuronal cysts in the brain for the lifetime of the host, right? So you at some point get infected, and you stay infected for your life. And these don't die. They actually have the ability to somehow maintain this dormant bradiozoite, semi-dormant, semi-dynamic phase. So this lab had actually done some previous work looking at chronic infection, and showing that in this situation, you end up with a loss of astrocytic glutamate transport GLT1, leading to this change in neuronal excitotoxicity. So they say leading to neuronal excitotoxicity. We'll dive a little deeper into that. Now, GLT1 can be regulated by neuronal-derived extracellular vesicles. We're going to all have to learn about what those are. And then here they wanted to determine if this cyst infection of neurons altered not only EV production, not only the production of the extracellular vesicles, but maybe the content. And then if perhaps the amount and the content of these extracellular vesicles might impact astrocyte function. And yes, this study, as they say, found that Toxoplasma cyst infection not only decreased the production of the extracellular vessels by neurons, but it also, we're going to see some evidence that it altered the contents as far as protein and the microRNA content. And then we're also going to learn a little bit about these extracellular vesicles containing parasite-derived proteins, secreted dense granular protein. And then they're going to go on and do some interesting work looking at pro-inflammatory changes that we're seeing, changes in astrocytic gene expression, and ultimately they're going to claim that this study demonstrates that the Toxoplasma cysts are going to alter neuron astrocyte communication by bypassing traditional immune mechanisms of recognition and leading to changes in astrocyte function. All right, so I think the first thing you jumped in with, the most successful parasite is key, because that makes this highly relevant. So, 1/3 of the world's population.
Yeah, it's a lot of people.
It really is. So if this is affecting behavior of 1/3 of the world's population, it may be interesting to know, sort of an us and them.
This has been an ongoing point of discussion in the field. Remember, we had John Boothroyd on TWiP a while ago, and he said he didn't think there was any evidence for overt changes in human behavior. There's some change in mouse behavior. They no longer smell the cat urine, and so they don't get afraid of them, right?
So clearly an impact on mouse behavior, right? Because we do see that normally, mice smell feline urine, and they're an aversion response, which makes sense evolutionarily, like get away from the areas where there's cat urine, danger. But yes, it has been demonstrated, I think, very clearly that once the mice get infected, they actually no longer have that aversion response. Mm-hmm. There also was I don't know if you remember, I think our listeners might remember the wolf study where they looked at wolves that had Toxoplasma infection and not. And even though there maybe are some issues with the concept of the alpha wolf, right? But there is this phenomenon where wolves leave their pack as adolescents, and then they sort of establish or take over a new pack, and much more likely, if you have Toxoplasma infection, to go ahead and follow that developmental path and become the leader of a new pack.
Interesting. Well, but they didn't deal with confounding variables, Daniel.
Sure, there's a lot of confounding variables there, right? That was a more sort of an observational correlate. So interesting. So a couple things to know about Toxoplasma. We talked about different ways that you might get infected. But one of the interesting things is that these cysts are actually intracellular. So typically you're going to have these intracellular cysts inside the neurons, in most cases inside the neurons. And we do know that there are some impacts, sort of obvious impacts. I mean, one thing which they point out based upon some other publications is that there is evidence that these cyst-infected neurons actually have a reduction in the dendritic spines and electrical activity. Right, so we're starting off less connections, less activity. So, sort of decreased neural function. And then I wanted to talk a little, because I think we're going to need some background here, is we're not only going to talk about neurons, but we're going to talk about glial cells, astrocytes. We're going to talk about non-neural cells. And I feel like you're almost more expert in this than me, Vincent, with all your time on This Week in Neuro. Is it neuroscience? Is it This Week?
This Week in Neuroscience, yes.
Yeah. And because there we've got a bunch of glial cells. And really, what is glial? These are non-neurons. So in the CNS, you've got about half of the volume is neurons, about half of the volume is non-neuron cells. And there's a number, right? There's the astrocytes, there's the Schwann cells, the oligodendrocytes, the microglial, the satellite glial. There's a bunch of cells. But the most abundant glial cell is actually the astrocytes. And they're important. They're sort of a support, a nourishment. They're important in promoting signaling. They help with transfer of nutrients from the blood to the neurons. So really an important. So here we've got our neurons, and we've got the most abundant non-neuron cell that we're going to be talking about. I guess the next thing we need to know is what's the story with GLT1 and glutamate? So we'll dive a little bit into that. So glutamate is the primary excitatory neurotransmitter in the brain. And astrocytic glutamate transporter, GLT1, is responsible for 90% of the extracellular glutamate uptake in the CNS. Okay, so sort of critical things. So
otherwise, if you don't get rid of that glutamate, it will build up in the synapse, and then you have dysregulation and excitotoxic states like seizures.
And I think that's critical, because now we get what was that excitotoxicity they were talking about. And if you don't clear the glutamate out of there, you get this high level of activity. And actually, you can actually start having seizures.
Yeah, so this is really important. This GLT1, and a lot of diseases have dysregulation of that.
Yeah, they mention a number, right? So if you have issues here with the astrocytes, you might have neurodegenerative issues, you might have issues with strokes, seizure disorders such as epilepsy. This whole glutamate axis is really critical, right? Now, this group had previously demonstrated that following Toxoplasma infection, there was actually a decrease in the GLT1 expression. All right, so let's think about what that means. Our listeners sort of think this through. So glutamate's in there. GLT1 is responsible for clearing that, maintaining it at a proper level. You get rid of GLT1. And actually, as that group had shown, that results in actually too much glutamate in these areas. So we previously knew that. So we knew that we know that glutamate is important. We know that GLT1 is important for getting rid of it. we know that Toxoplasma is somehow reducing the amount of GLT1, leading to too much glutamate. But the exact mechanism, GLT1 regulation, has yet to be identified, until now, right? That's that nature abstract thing. Here we show, right? So they're going to actually look into this mechanism. And they're going to look, and really ask this question, are the extracellular vesicles involved? So maybe a little background on what are these extracellular vesicles. So extracellular vesicles, I think this was recently being discussed on Immune, but a number of cells produce these extracellular vesicles, and they're membrane bound, and they've got proteins, they've got lipids, they've got different nucleic acids, right? So you get your DNA, your RNA, your miRNA. And all cells, we now realize that all cells are able to secrete extracellular vesicles. So this is sort of an ubiquitous phenomenon. It's something it sort of falls, when you learn your basics of like cells and extracellular, the extracellular space is actually, dare I say, infested. Like this is part of the whole way that cells interact among themselves and part of what's going on in that. Now, not only do our cells have to do it, but Toxoplasma, they can also. So parasites also have the capability of, as we're going to see, interacting with our EVs, but they can also produce their own extracellular vesicles. And sometimes those EVs, as we've seen in other studies, can interact with signaling pathways and cellular communication. So here's the sort of the ending of their introduction before we get into the results. The authors say, here we describe the ability of Toxoplasma infection to alter the production and content of extracellular vesicles from infected neurons and the resulting effect on astrocytes. Our results demonstrate a significant decrease in the concentration of extracellular vesicles and an alteration in the EV protein and miRNA content. EVs from infected neurons altered astrocytes, including an upregulation in inflammatory genes, a significant decrease in astrocytic GLT1 protein expression. And they go on to very humbly say, these processes reveal a previously unknown mechanism of parasite-induced changes in the brain that have the potential to broadly alter neurochemistry in the infected CNS. All right, so now let's see if this is really true. So let's go through the data. Usually I start by talking about the methods, but I feel like the methods are pretty straightforward and kind of tied in with each figure. So let's look at the data, shall we? So starting off with figure 1, which is entitled Primary Cortical Neurons Infected with Toxoplasma. So the first is sort of reading through the text. You realize they've got to figure out what's the best multiplicity of infection to use, where they're going to get infection, but they're not going to kill a lot of the neurons. Because unfortunately, too many parasites would cause rapid and complete neuronal cell death. So if you expose, to a high multiplicity of infection, you're basically going to kill all the neurons. So, as we see looking here, they're going to settle on an MOI, multiplicity of infection, of 0.5. And then we see actually some pretty nice experiments, sort of consistent with that. And not only consistent with that, but actually looking at they're doing these interesting staining. And I have to say, all the figures are beautiful. Dixon would have loved this, right? He was always a very visual guy. Yep. We've got DAPI staining, where they're staining the nucleus. We've got MAP2 staining, where they're staining the pericardia and the dendrites on the neurons. And I'd say that's beautiful. This MAP2 staining, it looks like the neuron should look. And then they do some other staining where we're going to actually see where the T gondii is. And so I think I made some notes here. If I go back and forth between my paper. So the DBA staining is actually going to show us the cysts, and then they're going to do some merge where we can actually see what's going on here. So really, really beautiful pictures, kind of setting us up for the future data.
It's a very nice cyst we see there, right?
Yeah, they're really confirmed. We picked a good MOI. We're not killing all the neurons. We're infecting, an MOI of 0.5. So we're infecting half of the neurons. Nice overlap between the cyst and the neuron. So everything that we think we should be seeing here.
The neurons are just beautiful, aren't they?
They're beautiful cells, right?
Yeah.
All right. And then the first claim, Toxoplasma infection leads to a decrease in the concentration of extracellular vesicles. And we have some nice data here and sort of walking everyone through it. First, we're looking at transmission electron microscopy. And we've got these different panels, and then we've got the quantification of the size of the EVs from the image. And I guess our statisticians are telling us that's different, but okay.
What do they think? On the picture, it looks the same to me. What are they saying is happening? What's their conclusion?
So they're actually going to say, and so B is the quantification of the size of the EM from the transmission electron microscopy images, and they're actually going to
same.
They look pretty similar. So I think the size, I guess I would say the size, I'm not impressed that there's a difference. But let's go to the next part where they're going to do nanoparticle tracking analysis. They're going to look at particle size and concentrations. Now we're moving all the way to D. And then you're going to move to, I think D is probably the best place to look at it. So D, you're looking at the sizes, and they've got them sort of quantified here. You've got PBS, you've got the uninfected, and the infected. And the infected, you're actually seeing a little bit of a difference there. But it's really the concentration where I'm seeing a big difference, right? The infected concentration is really dropping more than in half.
Yep.
Right. So that's probably the most impressive. So,
Absolutely.
All right, I'll take it. We are seeing a decrease in the concentration of the extracellular vesicles. And now we're going to move to, will tell us a little more about those extracellular vesicles. So there's less, but what about the content? And the first thing we're going to look at is the protein content. And we're going to do some principal component analysis. And then they're going to compare. And you see a clear separation when they do the principal component analysis. So very clearly changing the proteomics, the protein content, the different proteins that are in these extracellular vesicles. So we're getting less extracellular vesicles, more than half, and we're also getting a significant change in the protein that are in these. And they do isolate a few of them. So you're seeing a change in proteins involved in folding and double-stranded RNA binding, in cell adhesion binding, and synaptic vesicle endocytosis, regulation of the synapse organization, neuron migration. Pretty nice data, I would say. All right, so now we've established, right? We've got a good system. It's working. The cysts are the neurons. We're not killing them all. We've reduced the EV content by more than half. The EV number by more than half. We've changed the content when it comes to protein that's in there. And now we're going to look at miRNA. What is miRNA, Vincent?
MicroRNAs are small RNAs made by cells that regulate gene expression. They can bind to, say, messenger RNAs and inhibit their translation or degrade them. And so they're a way to regulate gene expression.
And this is, again, beautiful. You're seeing really clear separation when they do the principal component analysis. And then you're seeing particular microRNAs, a few of my favorites in there. I'm joking because I don't have any memorized favorite microRNA. Maybe you do, Vincent, do you?
Well, maybe the targets you might have favorite targets, right?
Exactly. Yeah, exactly.
Innate inflammatory markers and so forth. You would probably like some of those.
Exactly. Yeah, I think it's pretty hard to remember the actual microRNAs by their numerical designations.
No, there's no at all. They're just numbers. The only microRNA I actually remember is MIR122. I think that's the one. MIR122, I think it is. Yes. So that's a liver-specific microRNA that is required for hepatitis C virus reproduction. That's why the virus doesn't replicate anywhere else, because MIR122 is liver-specific. And that's the only one I remember because I talk about it all the time. Yeah, the other ones are not. But you have things like NF-kappa B, right? AKT and so forth that are targeted by these microRNAs. So they're all pro-inflammatory microRNAs that are being downregulating. And then they're downregulating pro-inflammatory and upregulating anti-inflammatory. So the toxo infection seems to be perturbing the inflammatory response, right? It sounds like it wants to preserve itself.
Interesting.
Well, it doesn't want to do anything. It's just that's the effect, right?
Yeah. Evolutionarily, how does this make sense? But yeah.
It's quite interesting because I suppose, and you know, at some point they could test this. If you didn't make these changes to these inflammatory, pro-inflammatory, and anti-inflammatory targets, I wonder, would toxo not be able to remain? You know, are these important for persistence?
That's interesting. Yeah. Is this like, are we worried about the impact on behavior? Is that just, by the way, is this just critical for the persistence of toxoplasma? Sort of. Right. Yeah. And it leaves, like, we have the technology now to kind of go through, like, sort of one at a time, taking these out. All right. So now the next is, so this is great, right? So we're sort of building our story. We've got great system, decreased extracellular vesicles, different protein content, different microRNA content, really sort of pushing things towards this pro-inflammatory excitotoxic milieu. But what's going on is, we talked about those extracellular vesicles that are being produced. Ultimately, they can be taken up by astrocytes. And yes, figure 5, we actually see the astrocytes are taking up the neuronal extracellular vesicles. And again, just beautiful. I mean, they have the great images, and then you see the quantification of this.
And you see that normally this always happens, right? Neurons make extracellular vesicles and the astrocytes take them up. It's a means of communication. Because if you look at the graphs, the percent astrocytes with EVs is the same in uninfected and infected, right?
It's pretty similar. I mean, the statistician tells us it's a little bit more, but it's going on, and maybe the statistician, but it's going on. It continues to go on. Maybe there's some increase.
Yeah, but what's different is what's in them, right?
Yeah. What happens once those EVs from the infected neurons actually end up in the astrocyte? So then we see that it actually alters the astrocyte gene expression. And what were we worried about? GLT1, that actually decreases. So we see a decrease in GLT1 protein expression. So sort of circling back into what we were after, like, how might that GLT1 be going down? Remember, the GLT1 has got to get that glutamate out of those areas to prevent the excitotoxicity state. And here we're seeing in figure 6 that we actually, EVs from infected neurons have changes in gene expression, and they decrease the GLT1 protein expression.
We don't know if that's microRNA based, though, right?
So we're not sure at this point. Like, is it the microRNA or is it one of the proteins, right? Because this is just those extracellular vesicles, which sort of raises, right? You know, reviewer 2 could jump in, right? You got to one at a time look at every single protein and every single microRNA, and then come back in 3 years. But then what they're going to do in figure 7 is they're going to take astrocytes, and they're going to expose them to the extracellular vesicles in vitro. And they're going to see, are there similar transcriptional changes as we saw with infection? Yes, we're actually going to see that. So you're sort of building a system so you can actually ask that next question. Like, let's just set this up in vitro, and let's start exposing them to the extracellular vesicles and see if we get what we see in an experimental system.
Right.
So pretty nice work, I have to say. I felt like they really walked right through.
I think it's very interesting, but of course, there's a lot more to do.
We're done. We're done now. We're done with science now.
Oh, that's right. Well, yes, this is an infectious disease, and we're giving infectious diseases a break, right?
7 years, Toxoplasma gondii can just run free, wreak havoc, and cause trouble. But no, just all joking aside. I mean, one of the reasons that I find this most interesting, really, there's a couple things here, right? One is, as we see more and more people with immune compromises, becomes clinically a more relevant issue, right? We definitely had a lot of problems during the HIV, particularly the early years before we had a highly effective antiretrovirals. Now that things are falling apart, this may become more of an issue again in those populations. We also have other populations that can be impacted. But I think really a profound question is this impacting behavior? Because I don't know, like I've sort of noticed lately, like there's some weird stuff going on, right? I don't know if you've been reading the news, and I'm trying to figure out if this is due to a little too much toxoplasma, cyst-containing cortical neurons.
Yeah. Well, we know that RFK Jr. had a pork. What is it? A dog or a pork tapeworm in his brain?
I think the rumor is it was neurocysticercosis, right? That it was it was the pork tapeworm, which is actually super common, right? It's a number one cause of seizures in the Americas, particularly like south of the Rio Grande.
It's interesting though, because seizures could be caused by an excess of glutamate in the synaptic space, as we learned here today, and so who knows what's going on there.
That's actually interesting whether or not there's a connection there, right?
Yeah.
And I think that's actually something that I don't think's really been looked into as deeply as one might consider. So you got a third of the world's population infected with toxo. We're seeing that the toxo infection increases the glutamate in those areas. So, should we be looking at trying to clear, which is a challenge, this bradyzoite, this sort of subdued form, which is metabolically active, which we're seeing. It's affecting the neurons. Would that potentially help with folks that suffer from seizures, or is there a connection with migraines and other sort of excitotoxicity states?
So when you get a toxo infection, the brain cysts remain for the rest of your life?
They remain for the rest of your life, and they're not completely dormant, as we're seeing. They're actually having an impact on your neurons, an impact on your astrocytes, an impact on your glutamate levels.
And patients can have seizures, and you said headaches as well?
So particularly when it comes to clinical attention is when a person's immune system has reduced, and they can actually have a significant number of neurological manifestations. Yeah.
Is there any way to eliminate the cysts in the brain?
I guess that's an issue. We don't routinely try to clear the dormant state. We're usually jumping in when a person develops active disease.
I see. Well, as we see here, that the presence of toxoplasma causes neurons to make EVs that have different things in them, which affect astrocyte expression. So they may, astrocytes could be involved in producing inflammatory substances that would help clear the toxoplasma, but those are being downregulated. So that may be a form of immune evasion, right? Which the parasite, maybe that's why the parasite is staying there. So maybe if you wanted to get rid of them, you could consider, increasing inflammation, although you don't want to do that in the brain usually, right, Daniel?
Well, that's actually one of the issues. Yeah, sometimes when someone has an acute toxo or we're treating toxo, sometimes we actually even have to use steroids so that the immune system does not become too significant, because it's a limited space. So, the inflammation can get us in trouble.
How many cysts would an infected person have in their brain? Many?
Many. So a significant number. When we often see this in the immunocompromised, there'll be multiple cysts.
And how would you diagnose a brain cyst?
So with toxo, someone has an acute toxo, or they have usually when we say acute, we're talking about a relapse because they were infected usually many years ago. They'll come in with sort of appropriate neurological, altered mental status or things suggesting pressure inside the brain. And then we'll usually do a scan. We'll scan the brain. And you can actually see these ring-enhancing lesions in the brain.
What kind of scan would you do? An MRI?
You could do a CAT scan. You could do an MRI. A lot of times, in today's world, we're doing both to get a better characterization. Yeah.
Wow. So this is cool because it may explain some of the symptoms, and maybe at some point could suggest some therapy, right?
I am, I'm thinking maybe it suggests a little bit more of a profound role of Toxoplasma in sort of diseases outside of those we normally think about clinically when we think about Toxoplasma. So, I always know Dixon was very entrenched in the toxo does not affect human behavior. So I wish he's still around so we could continue those emotional discussions. But yeah.
So the authors conclude by saying that so toxo changes the brain environment through these extracellular vesicles. They drive an infection-induced astrocyte phenotype that promotes inflammatory signaling at the expense of neurotransmitter uptake.
Yeah.
What would be the function of it, preventing neurotransmitter uptake by GLP-1? I don't know why it would do that.
Yeah, I'm trying to think. Human beings are a dead end host in general, right? Because we tend not to get eaten. But that historically wasn't always true. But I think a lot of the mammals that are being infected, if you could sort of slow them down a little, maybe I'm thinking of the evolutionary advantage of allowing them to be easier prey for those felines, right? Then you can help with the life cycle, because that's ultimately, the evolutionary niche where these cysts want to be. They want to be consumed by a feline. So if you think you've got a mouse and maybe the mouse just a little bit slower, it's a little not so sharp, not so bright, not afraid of your urine, not paying as much attention as it should, gets munched by the feline, kind of continues the cycle.
All right. So you're thinking maybe it's a behavioral thing, right?
I think, I do think, there's an evolutionary advantage if you can induce a behavior in your host that increases your likelihood of continuing your reproductive cycle.
Okay. Right. Well, that could make sense. That could be evolutionarily plausible, as we say, right?
And maybe in human beings, if they, dumb us down a little bit, maybe we won't require that, meat is cooked as well, and, some other things like that.
So what is the incidence of toxo in the U.S.? Do you have any idea?
So we have numbers from certain areas, right? Like, the high spots, right? Brazil, France, it's over 80%. In the U.S., it's actually probably half the population.
Just here, 11%. According to the CDC, it's 11%.
I feel like that's probably low.
Too low, yeah.
Yeah, okay. I feel like that's probably a low.
There's still a lot of people. Half the population would be a lot. 100 million, well, more.
Yeah.
What's the population of the U.S. now?
Approaching 400 million.
So 200 million people. Wow.
Yeah. So it's a lot. Yeah, it's a lot. Yeah, even if you go with that number of 10%, you're talking about tens of millions of people. So
how could so many people be so stupid, Daniel?
Well, I don't
actually. It's not so many. It's just a fraction that vote it's the Electoral College, Daniel.
Yeah. Yeah. No, I don't want anyone to get the sense of getting a toxo infection, it suggests that you're, cognitively impaired. No, I mean, a lot of people like eating their meat on the more rare side, and that's often the way people get this. A lot of people like their cats, and, it's another exposure risk there. So,
but in the U.S., cattle are no doubt screened for toxo, right?
I don't know if that's true. Should we check?
Yeah. Are U.S. cattle screened for toxo? I mean, they don't screen for prion diseases. Who knows, right? They are not. Cattle are considered relatively resistant to toxo. So you're not getting. But Dixon always said you'd get it from beef. In France, they like to eat raw beef, right? Beef Wellington.
Yeah, we think, I think pork and, I think lamb and things like that are sort of bigger.
Yeah.
So.
Yeah, it says sheep and pigs. Sheep too.
Yeah. Do people eat sheep? Don't they eat them when they're little? But yeah, so
lamb
pigs,
chops,
lamb,
yeah.
wild. Yeah, I guess we do eat. Yeah, we eat lamb.
And
right?
I don't know.
Lamb. They're so cute.
Well, many animals are cute. Mutton is the older sheep. You can eat older sheep. It's called mutton.
There's a famous place in New York City where I think Abraham Lincoln or someone of that ilk used to go eat, some mutton house, so.
All right, Daniel. Anything else before we say goodbye?
No, I think this has been a great paper. I love this stuff. It's in the ilk of the Last of Us. We got parasites controlling brain function. Maybe here a little more subtle than in that series, but exciting stuff.
All right, that is TWiP 264. You can find show notes at microbe.tv/TWiP. You can send questions or comments at twip@microbe.tv. Of course, that's where you would send your case guesses. So our next episode will be a reveal and a new case. And if you enjoy these programs, we'd like to have your support. Microbe.tv/contribute or parasiteswithoutborders.com. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone, be safe.
The Parasites Without Borders is a nice take on Doctors Without Borders, right?
Hey, who had the name first? No, I think it was them.
but did you come up with Parasites Without Borders, or was that Dixon?
Dixon and I, it was kind of a joint. We were probably driving around in his Prius, and we wanted to come up with something that really reflected our, the getting knowledge to people in places that need it the most, and not limiting our efforts to just one country. And I
think it's great. I love the name of the company. Yep. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. So Dixon always got upset when I thanked ASM for support. Because he said,
why is that?
Well, he wanted the parasite societies to support us, right? So I don't even know what they are. But
there is an ASP, and I think ASTM&H, and
yeah. So they should really support. But ASM, what the heck? ASM supported us from the beginning. They provide the hosting space for these podcasts, which is a significant expense. So
if they want to enter, the parasites organizations want to support us, so we'd love to have your support.
I think these science-based podcasts are good advertising for the society, that you're supporting the dissemination of knowledge, and not enough do it, which is unfortunate. TWiV gets support from ASM and ASV, which is great, and TWiP here gets support from ASM, but none of the other programs. The TWIM gets support from ASM, but none of the other programs have any of that kind of support. So we I guess we could be aggressive and pursue it, but we haven't done it. We love to have our listeners support us.
Yeah, that is great. We appreciate that.
All right, Daniel, I'm going to hand over to you the ending today. Okay.
All right.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.